Okay, so we go now to the last bit, which is my job, which is actually very easy after all this presentation. And let me start by explaining my tongue-in-cheek comment, which I started with, right? So last, how many? 6 or 7 decades of intense clinical development around psychiatry development, and nobody has found an answer to shorten the timelines. So if you have your hopes that in 15 minutes, I'm going to give you an answer, you're sitting at the wrong hopes. That's not what I am going to do. However, we will definitely look and see what can be done and what are the true pragmatic ways to address precision, which starts at the discovery level, precision, which will lead to identifying target, having right science. If we cannot convert that into creating more precise clinical development program, which is necessary for evidence generation, eventually leading to new drugs and new devices and new solutions to market, then we are going to miss a huge part of this question. So let's go into that and speak about some of those issues. These are some of my disclosures. So if you are a clinical developer and you have a self-flagellating tendency that you want to watch something which really scares you, you don't watch horror movies. You look at this graphic, because that scares the hell out of any clinical developer. And it tells you how long does it take and how difficult the journey is actually getting a compound from all the way from the discovery stage. And just to give you a number, this timeline has been increasingly over the last decades. Now it's pushing 13.8 years at this moment when we speak about from the discovery stage to getting drug into a market. And that's really, really challenging. And I'll speak in a moment about that. Over the years, this has not become better. It has gone worse. And there are reasons for that. Now, we spoke about that a lot has actually happened in this and on the preclinical and discovery side. The technology has actually allowed more precision there. Hugh can speak about there's more throughput there. He spoke about there are now technology finding more homologous, if that's the word you use, technology which allows us better translation to do so. So there's a lot happening there. Unfortunately, what you do there, and once you come to the stage of the clinical, that's where problems start happening for many reasons. We'll speak about that. One of the big reasons is that when you start going to phase 2 and phase 3, then you start feeling the heat of the realities on the ground, and we'll speak in a moment. But to wake you up, because this is second, a day, we're going to do this a little bit interactive. And the people online, you might not be able to contribute, but the people in the room will have a chance to participate. Anyone can take a shot? Months. 12 months, less or more? More. Less. Okay, that's a good hope. Okay, yeah. When you say more than 12 months, would like to give a number? The median. All answers are welcome because depending upon programs, it's not a real answer. Okay, I'll make it easy. 15 months. Now, if you want to do more precise phase 1 in which you should get your patients in, and that makes sense because you want to make sure that you're right from the phase 1, you have patients being part. How long this thing number would stretch from 15? What would be the next number for phase 1 only? Take a shot. From 15 to how many months it should increase? Sorry? 30? That's double. It's 35. How long do you think a phase 2 or phase 3 program take a month on average? Answer is approximately somewhere between 37 to 41 months each stage. And this is the scary part. If you look at the last decade and you say each of these stages have now become longer to do, which stage, phase 1, phase 2, or phase 3 on a medium has actually become more arduous and longest to do? It is not phase 3. It's phase 2. And that's something to talk about, and I'll come to in a moment about where exactly things have been, going not in the right direction. There's a real opportunity to address that. So we are supposedly living in the golden era of biopharma innovation. And I say this not because, there is a genuine behind the curtain feeling now, and that's true, that a lot more research is coming back to mental health after a decade and a half of journey when everyone is stepping out. Is the other way now. A lot is happening, which many people in the room should be aware is in public and not so public domain, but there's push to bring now CNS, specifically mental health, back in front again. Look at the number of distinct drug in development. 10 years ago, 3,200 drugs were in development. We are speaking about, after 10 years, 6,100 unique drugs in development. That's fantastic. That's good news, because more drugs in development means that patient will get better treatment. The pipeline is increasingly diverse. People are not just doing more of same. They're actually new targets. There are actually new modalities being tested. And that's reflected to just 14% of the assets use modality validated over the last 5 years. That's a good and bad news both, because the operationalization of getting new modalities in clinical development is far more complex, but it's good news for patients. So what has happened, because of all this excitement, there is so much money flowing into that. Just in past 3 years, 146 billion in life science venture capital funding. I'm not even talking about government money. This is just private money. That's the push. That creates problem. Why? Because when you have so much to do, eventually the funnel still comes to about working with patients and sites, collecting evidence. So R&D productivity actually has remained stubbornly low. If you look at the last 10 years, the spending, and that's inflation-adjusted spending, that increased 44% from 170 to 247 billion, but it did not change the average number of using US FDA approval. So you're spending far more money to still develop the same number of drug. And majority of that actually comes from the clinical development evidence generation problem. Now, industry doesn't like to talk about that, but unless we talk about that, we will never get on top of that, because industry actually needs to look at itself very hard in the mirror. Industry has not been in general the true champion of innovation. Now, it sounds counterproductive. This is not industry which is all about R&D innovation. Yes and no, because what happened that after, we're talking about 6 or 7 decades ago, after the serendipitous discovery of some drugs, everyone thought, let's make more of incremental value out of these drugs so that we can sell it more. 2 examples were SSRIs and atypical antipsychotics. Go and look. At best, there's incremental value with the next SSRI or next atypical antipsychotic. But everyone went down that route because there was a market in which, in spite of the competition, you can actually sell more of the drug by sometime little bit of better profile, safety profile, tolerability, in spite of having same value vis-a-vis the efficacy. So they kept on doing more of that till they found that the golden proverbial hen has stopped giving eggs, and that's they woke up. What does value means for creating for different stakeholders? Regulators and payers were always on people's mind, but what about patients? What about prescriber? That was not given thoughts, and we come to that in a moment. Creating more precision is not natural. And you must be wondering, why is that so? Because till a few decades ago, any big pharma company, there are always tension between science and commercial. Nothing wrong in that, that's good. But actually, it made a lot of sense commercially to give a drug to more number of people, even if it worked marginally in all of them, because you make more money. Now, I'm being absolutely brutally honest about that, because that tendency of throwing drugs without understanding that all patients may not help by that has not done favor to the industry in long term. And that's why there's a need for doing it more precise way. Biomarker development initially, and even I would say now, because it's so complex, has been unimodal or unicomponent. And Andrea spoke a little bit about that. While the science has started to allow you to use all different multi-components and do feature extraction, regulators are still finding their way around it. And it's a genuine problem, because how do you use the power of AI is to come either with an integrative or multiplex biomarker. That's a challenge even for regulators to address. So they're on a journey too. And finally, the desire to collaborate is a new thing for industry. It's happening now for the last decade or so. But earlier, there were a lot of pharma players chose not to publish their negative data, not to share. Thankfully, that is changing now, and that's a good news. So we have to talk about some time-tested strategy. And I put question mark, and you can see me smiling. If these were time-tested, they would have been working, right? Yeah. Well, some of these strategies are not respected. Some are ignored, but they remain valid. First is about how do you make sure that you have innovative trial design, to decrease the requirement of study participant. That's the holy grail. You'll have less number of participants. You take address and right effect size. Make sure you don't create just incremental good for nothing value. You will leave less number of trial participants. Things are becoming a little better there. Now, you learned about how Naval presented about that you can mine the data also to inform, which will allow you to design your trial, your statistical method. You can borrow, some of your control arm from real world data. Maybe one day it'll become synthetic, possibly in mental health, never, but you can do that. You can use Bayesian designs. So there's a possibility to have smart trial design. That's one. Simple patient-centric proposal. And I would love when Peter will come and speak about that, because my God, industry does not know how to do this. Let me take a quiz game again. How many endpoints on an average you think phase 2 programs have? Take a number. 10 or more. Very good, very close. 21. 21 endpoints in an average trial. And it's not just simple 21 endpoints, because some of them are dependent upon the EDC system, the electronic data capture system. We're talking about technology. We're talking about training. The sites have to deal with all of that. And how many of the companies, now they have started to doing it, they will not even talk to patients till they have some hope and confidence the drug would actually gonna go to the next stage. In other words, they will not invest into talking to patient early on. Thankfully, that is changing. And we'll talk a little bit more about that. Reimagine the point of delivery of care. What does that mean? In good old days, you go to a hospital and a site, you have relationship with them, they will convert data for you. Now, investigators are absolutely, they can't keep their neck above the water when it comes to their daily clinical care. In other words, investigators or sites are not interested as much in clinical research that they used to be. For example, US, which is a big clinical trial market, almost 83% of global programs have US sites. Out of all investigators, 50% of US investigators, they do one and done. What does that mean? They participate once in clinical research, they say, I'm done, this is not for me. Because it's not conducive for their day-to-day life. We also should think about that the site selection has to be more data-driven. Technology actually allows you to now model and look forward to say where you can go, rather than talking about just a historical guesswork approach about how to select site. Now, the point of delivery I was speaking about, there have been experiments now which are good. For example, the technology does allow you to do fully decentralized trial, hybrid trial, and there are interesting experiments to say, how can you bring patients to their natural environment? One example in the room is Boehringer Ingelheim, which recently announced collaboration with Walgreens. And many others have tried the point of cares like pharmacy to actually find patients, not just hospital, to see that there are smartest way to do that. But the problem goes much deeper than that. And what is that? Medicine started wrong. Medicine started wrong in the sense because it was top down. Look at any modern medicine, Greek, Indian, Chinese, doesn't matter. All started by saying wise men trying to solve problem for patients without asking patients what means to them. And don't get me wrong, a lot of human suffering was actually avoided, but it put disease in the center. We have been talking since yesterday all about diagnosis. We're talking about diagnosis don't matter, clinical symptoms matter, domain matter. But the patients go a layer beyond that, which is what is meaningful to them. And it's not only symptoms, and it's not only disease and diagnosis. This is an important part of precision. I'll come to that in a moment, because improving a measure of health through a therapeutic intervention is only beneficial if it matters to patient. That's such an important fundamental foundational ground truth to keep on talking about it. And for that, it's critical that you bring patients' input at each stage of precision, starting from asking as simple as that, does it really mean something to you? An example, in my past job, you will speak about that. We have been building drug for cognition. For example, in schizophrenia, many wise men came together, and with all respect, and they said, well, it must mean something, and that meaningfulness is functional improvement. How do you define that? Holy grail being, you may be able to go back to work, you may become functional. Patients, they even speak about, if I can remember things better, that's meaningful as well. If I can concentrate, that's meaningful as well. Well, I would love to go to job one day, but that's a journey. That's not the only function. So meaningfulness is a patient concept, and incorporating that is important by making sure that you have a framework of meaningful measurement, starting from exploring which part of the disease. Now, this is the precision piece of that. Before we think about the biomarker which I addressed, before you think about what endpoint, what part of the disease which patient does not want to become worse, wants to become better, understand that, investing into that, converting that into a concept of interest, making sure that will become an outcome which you can measure, you can statistically address that, and finally define and build up endpoints around that. If we are not doing that, then we are doing precision from the scientific direction only, and we are not connecting precision to precise patient's need. So what does it mean in practice? This means that we must have tools to connect, biomarkers, for example, to connect brain behavior and experience. We require symptoms, functional and meaningful endpoint. We require standardized measure, which we have been speaking about that. And the holy grail would be that eventually, and technology does allow that, at least in the space of digital measures, to go from more precise subgroup to n equal to 1 measures, which we eventually want to go. Regulators are trying to come midway, and they still struggle. I don't want to speak on their behalf, but when we speak about, for example, biomarker integration, there are 3 pathways, and this is not my slide. This is straight from regulators. Dr. Mary Thanhai presented in a forum which I just shamelessly copied, and she speaks about that all 3 processes are valid. You can do in the context of a drug approval processes. Pharma industry is not interested in doing that because it's very heavy lifting and they simply cannot do it on their own. You can do a completely separate biomarker qualification program, but that requires also many people coming together, and it's a fairly heavy lift. And people are trying to do that. I think, again, Martin can speak about how much of this has been exercised just in PRISM and try to go through that. It requires some heavy lifting. The pathway which is relatively easier but requires investment is scientific community consensus. And how do you do that? By making sure that we collaborate. Yesterday, people were talking about what can we learn from Alzheimer's, right? People were talking about the whole pathway from symptoms when they come after the disease has set in to predicting the illness, the making sure you look in the future, which Alzheimer's can do it now with the ATN framework. This would not have been done if 20 years ago, people would not have come together with a simple question like that. Can we put all the imaging data in some central place? The ADNI, the Alzheimer's Disease Neuroimaging Initiative Network, was simply that, and that set the tone. And you heard from FNIH, from Malaysia yesterday, how much needs to be done in our field. So I'm going to provoke this by saying that if we need to address this, we need to address this now, because this is a marathon, not a sprint. But for like any marathon, we have to start today. And I'm hoping that a lot what we discussed today would lead to some action about making sure. I have to shamelessly plug here a few things, because a lot is happening in this regard, which brings the right stakeholder together. Fondation FondaMental ECNP sitting together is an example. ECNP trying to bring the networks and thematic working group with industry is an example. UK Mental Health Initiative is trying to do the same. Similar efforts are happening with ECNP. We need to make sure we don't duplicate and we speak the same language. Thank you, and let's have some dialogue. So it's a very silent online community today. Yeah. And I keep on wondering whether it's my technology, because I really see a lot of people attending with no question. We're a little over, but we can take a few questions. So any question, comment for any speaker, for last speaker, yeah, go ahead and do so. Yeah.
So Vikas, thanks for that. I think you alluded to basically the global alignment on these principles and procedures. So what's your view on that? How should we arrange this?
Yeah, hard questions for me.
I bring the ball back to you.
Yeah, gosh. It's good to see that there is already effort happening in harmonizing. I mean, you are an example of that as you have been now speaking to other stakeholders. I think before we speak about harmonization, it's a good idea to go a step back and talk about. In mental health field specifically, I often hear this terminology when people they call it consortia fatigue. And I try to understand what do they mean by that. I think industry in the past has gingerly experimented and have learned from it, but I definitely believe that they can do better. And when I say industry, it's not just large pharma. I mean the whole environment in which biotech, digital companies, mental health digital companies, big pharma, academic institute come together to push. This starts from something as simple as that, what's the right question to ask using a right group of people? That's the first thing. Then about what efforts are needed, which is a people question, which is hard, you know that. And then third becomes the resources and then converting into actual effort. I believe the intentions are pretty right in this regard. The biggest question is actually the second, because resources and people, that's why efficiencies are important. For example, what ECNP does, if it competes with, let's say, UK Mental Health Initiative, that's a shame. Instead, it's making sure that the same pool of researchers and data and sites and investigators come together. So the questions, if they ask similar way, the data is collected similar way. We have to do longitudinal work. There is absolutely, and prospective forward longitudinal work. There's no shortcut to that. And that was the question which earlier asked as well. The time to invest into that is now, because unless we do that, we will not be able to find better biomarkers, a lot more needed, endpoints, a lot more needed. I spoke earlier about putting a rocket engine in front of a bullock cart. So imagine if you have all the tools to identify the right target, the right domain, and then you go and throw into phase 2 all that smart thinking into, I don't want to say anything hard about scales, let's say MADRS or HAM-D, which has nothing to do with the science and biology, you're going to fail no matter what. So that investment is needed.
Thanks.
Anyone else?
Maybe a last question because we're really late. I'm sorry.
Okay, so last question. Yeah.
Oh, well, I don't know whether this is a good question, but I'll ask it anyway. You know, there's a lot of big data, a lot of big talk. Where are the mechanisms coming from? I don't hear a lot about basic science. I don't hear a lot about engagement of basic science and the gap between what's happening preclinical in animal models and in vitro model systems and whatnot, and the identification of targets. I don't think the big data is going to give us the targets, which I think seems to be what y'all are saying. And if we just have enough patients and enough gene data or enough proteomics or enough this or enough that, that somehow mechanisms will emerge out of that or targets will emerge out of that. And I have my doubts, all right, or maybe not my doubts, but I think there needs to be more partnership with basic science, and I'm not hearing that part of it.
I think you're being too humble. Far too humble. I actually think we know a huge amount actually of what the targets are, the systems and circuits. It's the joining up between what we think is wrong in the human and what's going on in the animal. We have in the last 20 years in industry moved from what are referred to as animal models to doing things in a different way. So for instance, things like forced swim test. I haven't had a forced swim test done in a lab for 17 years. But that is often happening in academia. So there is a chasm between what academia thinks is going on in industry and what's actually going on in industry. But I don't think there is a chasm between coming up with the new therapeutic concepts and what we actually want to move forward. I think there is plenty there. That's actually why I put that slide up about what the targets were identified by Carol back in that, and Mark Geyer in that journal, in Psychopharmacology journal. Those targets exist, but very, very few of them, as a neuropharmacologist who's been doing this game for 35 years, very, very few of those hypotheses have been referred to as what I could say properly tested in the clinic. And that comes over time and time again. If you talk to any industry pharmacologist, they will have moved a molecule forward that either we couldn't get the right exposure in man, or when we went to clinic, then we got the wrong signal. Carmini asked me about the rate of failure in mental health. I was just counting it up in the last 3 years at Boehringer. We've had 7 phase 2 readouts, and only 1 of them was successful. In 3 of those phase 2 readouts, in a major subscale, the placebo patients left that study technically well. Placebo effect was so great that, and it was a clinical, this is the point of the load on the patient, how you do it. The way that the clinical trial was executed made the patients better. No, seriously, not on all parameters, but just a couple, and Boehringer will publish this data. So I don't think it's a matter about targets. It's actually getting to the point where we, as a clinical patient, payer, regulator community, can test the hypotheses we've all been trying to drive. And we've been talking about immunopharmacology, immunopsychiatry. We know that there's something in there, but we've got to learn how to measure it and give real world benefit for the patient and make sure the patient's involved in that process, rather than doing old things in preclinical science with the greatest respect.
And I have to, being on the clinical side of the development, have to agree with that. And that's actually true. That's where the problem lies. But I will not only be the naysayer. I think in the next session, we'll talk about that. This is a good time to go back to fundamentals. This is a good time to look at the good old nice practices about how do you design your study? What effect size do you address? What is meaningful? Are you making sure that your sites which are selected are truly interested? Does it mean something for patient? Is it science for the heck of it? Or does it create some value for all the stakeholder? Those are the questions to address. And those foundational time-tested strategies remain valid. We have just become complacent about them. So let's talk about, in the next session, and I'm really looking forward to hearing some patient perspective too. We're running late, so we have been told that we need to shut down, and that's what we do. And we'll take a coffee break now for 15 minutes. No, 10 minutes, Guy. Okay, so 10 minutes, and 10 minutes, we are back. Thank you, everyone. And thank you to the speakers. Thank you so much.